Okay, welcome everyone. A hearty welcome to all of you on this warm evening. My name is Rajgopal Patil, and I am the Secretary of Institute of Natural History, Education and Research, Pune, along with Maharashtra Vruksha Samvardhini, and in association with the Department of Botany, at our Jhunjhunwala College, Mumbai. We are very happy to have you all for this online session this evening. Okay, so some housekeeping instructions. First and the most important, let's keep our microphones on mute when we are not speaking. We want to hear the speaker and definitely not the sound of background traffic or someone's pressure cooker, and most certainly not the dialogues of Tharla Tarmak playing on TV. Of course, as you listen to the talk, some questions will pop up in your head. Hold on to them. We will take them at the end of the questions parade or wandering wall. Put them in chat box if they stop you from focusing on the talk. So let's get started. Picture a hidden world beneath our feet, where tiny soil microbes work tirelessly, weaving an intricate relationships that shape the diversity of plants and the health of our ecosystems. Today, we are going to uncover the fascinating stories of these unseen heroes and explore how they influence the vibrant mesh of the life around us. To take us into this mysterious world, we have the renowned scientist Dr. Mukund Deshpande. I request Ajit Vartak sir, the Secretary of Maharashtra Vruksha Samvardhini, to introduce our esteemed speaker.
Good evening, everyone. I am delighted to introduce our guest speaker today, Dr. Mukund Deshpande, a renowned microbiologist and a dear friend for the past 40 years. Dr. Deshpande obtained his MSc in microbiology, PhD in biochemistry, and his extensive work in the area of fungal biology, especially fungal differentiation, earned him DSc in microbiology from uh, Savitribai Phule Pune University. Since then, he has been working extensively on the use of fungi and fungal products in human healthcare and agricultural biotechnology. He successfully completed more than 35 research projects funded by national and international funding agencies. To name a few as Indo-Swiss program of DBT, Indo-Belarus program of DBT, Indo-Mexico program of DST. Dr. Mukund Deshpande has to his credit more than 170 research papers, reviews and chapters, 9 patents, 12 books, and a number of popular articles. He is a highly respected researcher with an impressive record of 7,900 citations for his publications, and his H index is 49 and R10 index is 90, showcasing his significant impact in his field. 21 students obtained PhD degree under his supervision. He developed three technologies useful in agriculture for the pest and pathogen control and licensed them for his own startup, Greenvention Biotech Private Limited. Dr. Deshpande is elected Fellow of Maharashtra Academy of Sciences and the Society for Biotechnology Advancement. He was also recipient of Department of Biotechnology Overseas Associateship in 1995 and Commonwealth Science Council Fellowship in 1998. He has received many awards for his work and service. Some of them are Professor K. Natarajan Memorial Lecture Award by Mycological Society of India in 2013, Lifetime Achievement Award 2014-15 for his work in both basic and applied aspects of chitin and enzymes which can modify fungal and insect chitin by Indian Chitin and Chitosan Society and MS Marshall Marine Products, Tamil Nadu. Dr. Deshpande is Chairman, Industry Advisory Board, Amity Institute of Biotechnology, Amity University, Gurgaon from 2024, member of SAC Steering Committee, DBT NECAB Center, AAU Jorhat from 2014. Uh, Beyond his scientific achievements, he is also a passionate nature photographer, delighting his friends each year with a beautifully crafted photo calendar of his nature photos. Additionally, he has a keen interest in philately and has contributed to the field by authoring one of the first articles on mycology and mycotechnology on postal stamps in Current Science, India's premier scientific journal. With this short introduction, now I request Dr. Deshpande to deliver his talk. Dr. Deshpande.
Thank you, Ajit, for giving introduction, which is quite extensive. Though you are saying that is a short introduction, this was not short introduction, but anyway, and this has increased my pressure also and nervousness because all the mm, people who are working on the plants and diversity, they are sitting in the audience. So, shall I share my presentation?
Yeah, please, sir.
Now, I would like to start with my association with Agarkar Research Institute. I joined Agarkar Research Institute as an MSc student, MSc in microbiology. That time, that center, Pune University Center, was in Agarkar Research Institute, and mine was the third batch of microbiology of Pune University. That time, Dr. Godbole, Dr. Y.S. Kulkarni, Dr. Vijay Rao, Dr. Pauri from NIV, and few others used to teach us. Basically, then MSCS, we used to know that time, not Agarkar Research Institute, was a research institute, and therefore research culture was inculcated in me. And whatever Ajit has read, that is because of my association with ARI that time. There are few other departments like botany, plant breeding, paleobotany, and chemistry. Dr. Vartak, sir, used to work on the first floor in the main building. And whenever we used to go to library, that time we will see that always he is on the microscope or he is doing something in the laboratory. He was unassuming scientist, greatly respected by colleagues and research students and MSc students like us. We never asked any favor from him, but we were so confident that he would help us unconditionally. When Ajit asked me about giving lecture, I immediately agreed. Though I did not get an opportunity to work with him, I could go through some of his papers about Devrai. He used to say sacred groves are nothing but the treasure troves. Today I am trying to connect my understanding of microbes, especially soil microbes, with his observations of above ground plant diversity. This is my tribute to Dr. Vartak, sir. How to change this one then next? Okay. Yeah. So it is all about the unseen majority of soil microbes, which according to me and according to number of microbiologists, that these are nothing but the drivers of plant diversity and ecosystem. Of course, this is hard to believe, but I'm sure that at the end of the lecture, you will also find that yes, microorganisms are certainly useful organisms for this diversity. Now, when we talk about diversity, uh, one should know that how many species exist on Earth. For example, bacteria, 43,000 bacterial species are known, but the estimated number is around 1 trillion. Fungal organisms, with which I have done lot of work, 1,50,000 known species are there, and 51, that is 5.1 million estimated number is there. Maximum number is of insects. It is estimated number is 6 million, and 8 lakh insects are known. So these are the different levels of the species which are present on the earth. So when we talk about the bacteria, because we want to see that the unseen diversity, around 43,000 formally named species are there and which are in pure culture. I'm not talking about the uncultured organisms, and their physiology is also has also been studied. The species definition somehow now in the recent world, everybody is talking about the molecular approach. But I would like to give one example, which the uh, Dr. M.C. Srinivasan, who has done a lot of work on fungi and bacteria, used to say tell us that Streptomyces and Chenia, These are the two genera when he was working with Dr. Tirumulachar. Molecularly, both the genera are, it is not the two genera. Molecularly, this is the one genus, that is Streptomyces. He had lot of discussion with Dr. Hopwood, the actinomyces person, and that according to Dr. Srinivasan, that the Chenia is the organism, that is the actinomycete, which has scleroshell bodies present. So this is the phenotypic difference, but unfortunately nowadays we are not talking about the phenotypic differences, we are just talking about the molecular approach. So this is the way. Initially, when we studied, we used to work only on the phenotypic or the biochemical differences, but nowadays it is because of the molecular approach. But okay, to cut the story short, we can say that now the species definition is based on the molecular aspect. Uh, so according to this molecular studies, we can say that 1 trillion species of microbes are present on Earth. In case of fungi, as I said, that the number of fungi is 1,50,000 known or named organisms. And usually we find that the six fungal organisms are present with one plant species. So with this type of number, we'll find that the microbiology is certainly important. And the person who has made microbiology significant in molecular world is Carl Booth. What he used to say that, so like it or not, microbiology is going to be in the center of evolutionary study in the future and vice versa. And his concept is not only this organism per se, he is having the concept of holobiont. That means the microorganisms which are present in the host, and the host DNA and the microbial DNA is togetherly increasing or togetherly going one generation to another generation. And here you can see the list of different organisms where we can see that number of bacterial species are present. In case of plants, for example, Phylosphere 252 bacterial species are present, while in as an endophyte, which is now is a key uh, element in microbiology and especially in the medicinal world, that 77 endophytes are known from bacterial endophytes, but fungal endophytes are more. In rhizosphere, you can see the 30,000 bacterial species. So in case of the plant diversity, you will find that the plants, they are always growing with the microorganisms. Here it is, one example is given about the microalgae, and you will find that the both host genome and microbiome can be transmitted through generations, and thus they maintain the unique properties of the holobiont, that is the morphology, then development, behavior, physiology, and resistance to diseases. And this is one of the important aspect, that the disease resistance is always in the plant is brought by microorganisms. So when we talk about soil microorganisms, how many soil microorganisms are present? Now, in the surface soil per hectare, we can find around 1 to 10 tons of bacteria uh, in the surface soil, and these bacteria community or the microbial community are all having the from functional point of view, they are working as same everywhere, but some additional properties or characteristics they have which can make them the unique organisms. So the first question anybody will ask is, microbial diversity is really greater than the microbial or the we can say that the plant or any animal diversity? Answer is yes. Why? Because metabolically microorganisms are more diverse. In case of plants, you will find that the plants, for example, they generate energy through photosynthesis. In case of bacteria, they can do photosynthesis, as well as they can use they can grow saprophytically also. Then microbial biogeography, everything is everywhere, and most of the microorganisms are present everywhere. And microbial dormancy, this is quite interesting, that the viable bacteria have been retrieved from 500,000 years old permafrost, and so we can say that they have good dormancy as far as their eventually evolutionary behavior is concerned. So microbial evolution is always fast because the generation time is fast and rapid rate of mutation is there, and because of mutation we can see the evolution. Now, when we talk about soil microbial diversity, does it really play any role in resisting and restoring degraded ecosystem? This is one of the recent papers which I would like to quote as Plant Soils, this 2024 paper. How we assess first the degradation or land degradation? We can assess by satellite imagery, maybe remote sensing, ecological models, computational models, and of course, if I don't say artificial intelligence, then people will say that I am old generation person. So it is artificial intelligence also is important. So what are the different microbial technologies are involved in the resisting and restoring this degraded ecosystem? One is the microbial inoculant. We are going to talk about this one because the basic agriculture is dependent on these microbial inoculants. Then synthetic microbial communities. It is not only one organism. We can have the mixtures of organisms which we can use, and microbiome engineering. And this is the last aspect which I would like to highlight as a future of microbiology. So you might find that this is very primitive, but I thought that maybe I should start with this one and then go little complicated things little later. So there are four different uh, aspects. One is the carbon cycle, nitrogen and phosphorus cycle. These three cycles are important. And one group of fungal organisms, which nowadays also it is highlighted in agriculture community, is the use of mycorrhiza. And mycorrhiza are not only important in agriculture, they are also important in the restoring different type of plant diversity. So I'm not going to give all the details, but say, for example, carbon cycle, there are different bacteria, nitrogen fixing bacteria and other bacteria, even pathogens, etc. Mycorrhizal fungi, they contribute around 50% of the uh, carbon cycle, or the presence of carbon. Photosynthetic algae, for example, they are involved in CO2 fixation. Chemoautotrophs also there, and bacteria are having both types. They are going for the CO2 fixation by photosynthetic approach. Then in the respiration, that they have the TCA cycle, and in fermentation is also important as far as the saprobiotic organisms are concerned. Under anaerobic condition, you will find there are different microorganisms. So name the environmental condition, you will find microorganisms are contributing there. Initially, actually, when I did my MSc, after MSc, I was working in one of the projects on the methane production. So, I mean, this is quite close to my heart, because that time I identified few methanogens which are important in the vitamin B12 production. And that was my first publication, I can say, from MSCS, or from Agricultural Research Institute. So, plants, for example, they are either consumed by animals, or maybe because of their death, soil organic matter is there, which is increased because of the plant death. Then we will find that the methane production by methanogenesis, or by methane oxidation, we can go finally to the microbial biomass. Then carbon dioxide, that is coming from plants and also fixed from the plants. Same thing, CO2 is coming from animals, and bacteria are also important in decomposition of organic matter, etc. Now in soil, it is not only carbon, but nitrogen is also important, and the CN ratio. Most of the time, what we do in the laboratory, that we do not consider the CN ratio. We consider individual nitrogen source or the carbon source. But when you consider both, then you will find that you can see the real picture of the growth of the different organisms. For example, in fertile soil, 3.5 to 4 is the carbon percentage, and maximum is 10% carbon has been seen. But CN ratio, we can see that in legumes and farmlands, for example, 20:1 or 30:1, or in microorganisms, usually it is 4:1 to 9:1. So CN ratio is important because the CN ratio decides the soil fertility level, and arid soils, we can see that the CN ratio is low. Now the second cycle is nitrogen cycle. Again, these steps which were, I'm just mentioning that nitrogen fixation, nitrogen assimilation, ammonification, nitrification, denitrification. These are not the steps, individual steps, or in this order. Maybe individual steps are all the time, they are simultaneously going on. So nitrogen fixation is one of the important aspects. Again, I here recall that Professor V.P. Bhede from Agarkar Research Institute, he was principal of agriculture college, and then he was working in in ARI after his retirement. He used to teach us, and that time he has started the laboratory where for the first time azorhizo cultures, that is azorobacter and rhizobium. One is non-symbiotic, another one is symbiotic. These cultures were uh, produced, and for the first time they were used in the field for the nitrogen fixation. This is some additional thing. Azotobacter is one of the useful organisms for nitrogen fixation. Growth, it can also produce growth hormones like indole, acetic acid, etc. It is also important in phosphate solubilization, plant disease management, or maybe pesticide degradation, heavy metal metabolism, etc. And usually its efficiency is also quite high, that 20 kg of nitrogen per hectare per year it can fix. So these organisms are certainly contribute in the nitrogen, and finally it is not only the nitrogen, carbon cycle, nitrogen cycle, these are, they contribute in the plant development. In the nitrogen cycle, we just now I mentioned about nitrification, denitrification. So these aspects are also important. And again, there is a list of organisms that Pseudomonas, Bacillus, Microbes. So these are the organisms, or Acrobacter fisheri. I started my PhD work on Acrobacter fisheri. It is the bioluminescent organism, and present in the fish, and fish can search food in the light of this organism. So I worked on with this organism on nitrite reductase and nitrate reductase, these two enzymes. So these are the organisms which are important in the nitrogen cycle. This is one of the important organisms recently that we identified is Gluconoacetobacter. This is an endophyte, first isolated from the sugarcane, and afterwards it is reported in different crops as coffee, ragi, pineapple, and rice. And besides nitrogen fixation, this bacterium also produces different plant growth promoting substances. So from this list, certainly you will find that yes, these are the organisms which are important for the plant growth. We are not come to the shifting of the plant species or something like that. That one we'll see a little later. Mycorrhizal fungi and the phosphate solubilizing bacteria are important in the phosphorus cycle. Again, there are different organisms like the plant growth promoting rhizobacteria like Klebsiella, Pseudomonas, Escherichia coli. So these are the organisms. They certainly contribute in the phosphorus cycle. I'm just going a little hurriedly because these are the known facts, and these are quite common facts, especially in agriculture. In case of mycorrhiza, you will find these are nothing but the extension of the plant roots. And that is the idea of this mycorrhiza. Actually, mycorrhiza are not biofertilizers. They are physical extensions of the plant roots, and they can increase the longevity of feeder roots. They can increase the surface area. They can be important in the selective absorption of immobile phosphorus, zinc, or copper, or maybe other mobile elements. it can enhance the water uptake. And these organisms, you will see later that how these organisms are important. Mycorrhizae are important in the changing the scenario in the field. That means the change in the plant diversity. So these organisms, this is important for the agriculture people, that nitrogen fixing organisms which are free living and the symbiotic organisms like rhizobium, etc. Phosphorus mobilizing organisms, then biofertilizers for micronutrients like bacillus, and PGPR bacteria like pseudomonas, and plant growth promoting fungi like trichoderma, metarhizium, etc. So this is the total scenario of any agriculture field, and these organisms certainly contribute a lot, especially in agriculture. And the same thing is true when these organisms are also important in the forest development. Uh, just first example which I would like to take, that about the mangroves, that plant growth promoting bacteria are important to increase the reforestation of the damaged forest of artificial mangroves. So mangroves are increased with the help of the plant growth promoting bacteria, and that is the well studied example which um, which is about the plant growth promoting bacteria, which can be like nitrogen fixing bacteria or phosphorus solubilizing bacteria, or bacteria which are having the it can produce phytohormones, etc., or even the biocontrol organisms of phytopathogens. So this is the first example where we can say that these organisms can be used for the reforestation of the damaged forest. Uh, this is again quite recent concept. And if you read the title of this publication, that the plants as the extended phenotype of endophytes. So actually the number of bioactive molecules are present in the plants which are medicinally important. And now the concept is that these molecules are really produced by the plants, or these molecules are produced by the endophytes. Are endophytes crucial for plant existence? So that is the second thing which is important. And according to me and according to number of observations, we can see that these organisms are certainly contributing a lot in the diversity as well as the plant existence. Now, for example, what are the different types of plant-associated microorganisms? We can see that the rhizospheric or rhizoplanic microorganisms are there, that everybody knows. Phylospheric also know. Endophytic organisms, and these organisms are either facultative or obligate or systemic. So these organisms are present in the plant. There, that is mutualism. Both are happily staying together, and number of times we can see the horizontal gene transfer. We have worked on this aspect, and these organisms are certainly contributing in the plant development. Also, it is it is also possible that these microorganisms or the fungal organisms which are endophytes, they are taking some traits from the plant. That one we'll talk about. So you will see that the number of endophytic organisms are present, and we find that the well-known examples of the Texas plant. Texas plant produces the anti-cancer drug, which is a billion-dollar industry, and you will find that there is fungal endophyte, the taxomyces, which can synthesize this taxol. So that is the first example I can say that the endophyte is being used for the production of the drug or for the chemical which is produced by the plant. There are total 66 bioactive molecules which are produced by different fungal organisms like Aspergillus, Fusarium. All these are endophytes: Polytotrichum, Epicoccum, Tramatis, Taxomyces, and number of others. So you will find that this trait from the plant, it has gone to the fungal organisms. Now, but there are certain fungal endophytes, like Curvularia, for example. It can affect the metabolism of the plant, where the, for example, vinblastine production is affected because of the endophytes. So this type of endophytes also there, which can increase the production of the bioactive molecules from the plant. And this is the point where I thought that maybe I can connect my research activity or my research work with the what Dr. Vartak sir has said about the Devrai. This is my work actually from my laboratory when I was in NCL, that this is the horizontal gene transfer from plant to fungus. It is possible that from fungus to plant, horizontal gene transfer is also possible. But here we are giving, I'm giving another example about the insecticidal traits from Annona, that is Sitafal, uh, can be transferred to the endophyte, that is Metarhizium anisople. Metarhizium is known as insect pathogenic fungi or entomopathogen, and its virulence against this insect pest is because of the insecticidal traits. Of course, Metarhizium has its own characteristics, but because of these insecticidal traits coming from the Annona, it is becoming more and more virulent. And that one we have seen, and we could publish this paper in Basic Microbiology Journal. Now, can soil microbiodiversity influence plant metabolites and life history traits of rhizophagous insects? So, there is one example which has been given, that these uh, uh, insects, or phytophagous insects, usually they affect the biochemical composition of the plants. Plant metabolites can influence major life history traits of these insects, and eventually we'll find that the rhizosporic microorganisms are an important biotic factors modulating plant metabolites. So, for the control of insect pest, you will find that the plants have some metabolites, but these metabolites are controlled by the rhizosporic organisms. So eventually you will find that the rhizosporic organisms are controlling both plant metabolism and as well as the insect pest control. So there is example of the oilseed rape, that is brassica, and cabbage root fly, and you will find that the rhizosporic microbial communities are important in plant defense. So when we talk about is there any shifting of the diversity or something like that, and you will find that how this microbial diversity drives multifunctionality in terrestrial ecosystem. So there are different examples which I have given here, but everything cannot be just discussed right now. But you will find that the biodiversity of rhizobia uh, is important, and these rhizobia are host specific usually, but they can shift the diversity of forest, tropical forest. For example, with the legume trees, and the organisms which are important are rhizobium, cynorhizobium, mesorhizobium, bradyrhizobium, and azorhizobium. These are the different 44 species are involved in the shifting of these tropical forest nature from in the leguminous plants. Uh, Number of studies have been carried out using different bacteria and fungi which are present in dry land, and in dry land, especially the fungal diversity is more useful because of their tolerance of than the more tolerance than the bacteria. Now, when we talk about individual microorganisms, it is not the individual microorganism per se. There are a lot of interactions are going on, and there is micro interaction, whether it is soil type is also influencing that one, plants also they influence, or the resources availability, maybe for carbon metabolism, nitrogen metabolism, and all other phosphate solubilization, etc. Resources are important. So these are the different factors which are involved in the evolution of the microbial community in the soil, and eventually it is important in the controlling the diversity of that soil. Now, this network of co-occurring microbial genera in rhizospheric samples, of course, so many times I have shown this in different talks, but I really understand this type of network. But it is quite impressive. You will find that in most of the places, lot of interaction is going on. It is not the single organism which is important. For example, azotobacter. Most number of times we think that it is only for nitrogen fixation, but no, it is also important in the phosphate solubilization. Same thing is rhizobium. Rhizobium is symbiotically fixes nitrogen in leguminous plants, but it is more used as phosphate solubilizer. So these are or Pseudomonas, for example. Pseudomonas is the organism which is again involved in the phosphate solubilization, and also it is important in the control of the plant pathogenic fungal organisms. So this is some sort of devrai, we can say, that they are the undisturbed forest in Europe, in European country, and right from 1838, they have kept this undisturbed forest, and the people have seen that what are the different habitats are present and how these habitats are interacting with each other. Of course, the microbial diversity is also coming in this. So it is that is European one. Here we have the Devrai, and I had an opportunity with going to one Devrai with Ajit, and I have seen this Devrai. So the important point in case of Devrai that uh, there are organisms which are present in the Devrai, and there are organisms which are present in agricultural land. For example, some bacteria or some fungal organisms, same for Metarhizium or Beauveria, these are the insect pathogenic organisms, or Trichoderma, which is the fungal controlling organism. So these organisms are present in agricultural soil. But we can go to Devrai, and if we isolate, say, Trichoderma, Metarhizium or Beauveria, or for that matter, any organism from Devrai, Devrai is having the virgin diversity, and because of that diversity, they are there. You will find that the organisms are interacting with each other in nature. Means insect pest is present, and insect pest controlling organism is also present in Devrai. This thing doesn't happen in agricultural land. So if we isolate Metarhizium, Beauveria like organisms from Devrai, they will have lot of potential. Rather, they will be more virulent as compared to the isolates from the um, agriculture field. And that was my main idea. And actually, we wanted to start some work on the microbial diversity, how this diversity is present. Actually, long back, I gave one program to DVT about biodiversity park. And in that biodiversity park, the concept was that you should have pest, pathogen, crop, everything at one place. So these organisms, they will be more interactive, more virulent to each other, and such type of diversity park is there. Then we can isolate different organisms which are having that sort of more different types of traits, insecticidal traits, or maybe antifungal traits, etc. So that was the idea. So Devrai, or maybe for that Devrai or any forest which is undisturbed, you will find that the microbial community is more uh, productive, I can say, physiologically more effective uh, in such type of undisturbed forest than the other places. So you can see that there are different types of connections. Mycorrhizal fungal organisms, they are important uh, because they are around roots. Leaf endophytes, they are also important, as I mentioned about the Metarhizium anisopli, which we have isolated from custard apple. So that sort of endophyte is also important because having that sort of insecticidal traits. Now, why leaf endophyte? what is the difference between the leaf endophyte and the maybe root endophyte. So the organisms which are isolated from the leaf as as an endophyte, they can work against the pest which is attacking or leaf eating insect pest, etc. And the another aspect is that when we talk about the root endophytes, certainly they are effective when the soil paste is there. Now here is one example which I would like to give is that people have studied the diversity of evergreen forest and deciduous forest. And in that one, they have seen that its diversity is different. In evergreen forest, there are different types of bacteria. Of course, this is the new classification, the gamma proteobacteria, clostridia, acidobacteria, etc. So these are the present in and deciduous trees. Again, you will find that the dominant diversities of the gamma proteobacteria, alpha proteobacteria, clostridia, acidobacteria, and beta proteobacteria. And when such type of diversity is there, that is there any possibility of using this diversity to change the total scenario of the forest? So it is possible, especially the mycorrhiza, they are important that they can change the diversity. For example, the mycorrhiza is associated one type of organ. This is symbiotic association, is one type of plant species. And when that plant species is when becomes invasive, so when we take that sort of plant species in the another field along with the mycorrhiza, that mycorrhiza certainly affect the diversity of that area. In this case, we can say that the habitat modification is there, then nutritional modification, pollination, competitive release, and predatory release. All these aspects are seen. These are the different mechanisms which are involved in the changing the diversity. Or habitat modification is the limiting resources, increase habitat complexity, functionally replace and native species. So this is according to me is important that how this happen that they can change the native species by just introducing the mycorrhizal organisms. On the other hand, there are three types of facilitation: the novel, substitute, and indirect, in which we will find so these are in the picture that this is given that, and just not going into those details, we will find that the native mycorrhiza fungi will associate and spread with introduced plants or by declining the pest attack on native plants. So this is the way it can happen. Most of the time, the microbial diversity will control the pest and pathogens and thus can protect the native plants. Or sometimes we will find that the microbial diversity, if increased in a different way, maybe some plant species are suppressed and some plant species are going up, and then we can eventually we can see that there is a difference in the forest. Mutualism is important for plants in new habitats to become naturalized species. And in case of an animal-mediated pollination and seed dispersal, mutualism is also important. And in all these things, we can see that the symbiosis between plant roots and microbiota is important, and that association can shift the plant diversity. And as I mentioned, that mycorrhizal fungi are important, and in addition to microbial fungi, nitrogen fixing associations between legumes and rhizobia is also important. Now this is the highlight of the talk, I can say, that conversion of tropical forest to rubber plantation. Now hereby this is again the metagenomic analysis that you will find that there is the relative abundance of different type of microorganisms or bacteria. And in case of another aspect, uh, forest, there's a rubber, for example, plantation, you will find that the different types of microorganisms. And if you increase the proportion of these organisms, of one type of organisms, there is a possibility of eventually we can shift forest to the rubber plantation. So that was the paper which was published in, I think, in 2017-18, and it was shown that yes, we can shift the forest from one type of forest to the different type of plantation. I mean, it is difficult to go through all the names of the organisms, etc. But this type of shifting can happen. And that's why this shift or analysis, molecular analysis is important at this point of time, because molecular analysis can identify the unculturable diversity. 90% is unculturable diversity is present in soil. So whatever we are seeing is just not more than 5% diversity, culturable diversity. So because of the molecular aspect, that is metagenomics or the functional genomic mm, analysis, we can see the different types of diversity. And in addition to the cultural diversity, this unculturable diversity certainly influence the eventually the plant diversity. Now, this is again just some sort of repetition, I can say, that this is one recent review that, not very recent, soil microbiome manipulates the resistance in plants and to the above ground insects. And if you remember, initially I mentioned about that how this microbial diversity is increasing the resistance of the plant towards the insect pest. So, same example in a different way. So now, on one side, we are saying that yes, microbial diversity is important. So can we have this diversity changed in the soil? And by changing the soil, can we change the, what should I say, that the nature of the soil? And for example, if you want to eradicate some pathogenic organisms, you can have the different fungicides, etc. Or and then you can remove all these pathogens. But it is likely that if you remove the total pathogens from the soil, then you will find that some other pathogens are taking over. The best example which I would like to give is that about the BT cotton. BT cotton is developed for the resistance to one insect pest, that is the Helicoverpa armigera. And because of BT cotton, we will find that the Helicoverpa armigera, that pest, is not there. But as the pest is, that major pest is not there, it is likely that there is a shifting of pest. And there is now, we find that in BT cotton, we can see that either millibug or thrips, they are now coming up. So this shifting of pest. So instead of shifting of pest, can we avoid the shifting of pest? So yes, we can have the suppressive soils. And this is one new aspect which one of my friends from Australia, this Professor Gupta and his colleagues, they have developed that we can have the shifting of pest approach. In it, we can stop the shifting of pest or pathogen by making the disease suppressive soil. So how to make this disease suppressive soil? So there are different organisms that from Burkholderia, Xanthomonas, or maybe uh, mm, other organisms like Bacillus, Pseudomonas, which are the fungal pathogens, or maybe Trichoderma or Metarhizium. All these organisms they are using to make the soil rich with this type of organisms, and they are suppressing the pathogens like Rhizoctonia or Sclerotium, that is a root infecting pathogens. And such type of disease suppression, this is the major program in Australia now, that you can have disease suppression, and thus uh, it is not necessary to use different fungal fungicides, etc. And that soil will be more useful as far as the mm, crop yield, agriculture crop yield is concerned. So this is one new aspect, which is again the change of the microbial diversity in the soil to make the diversity useful for other people. This is one of the interesting projects which I had about the petroleum contamination. It was seen that near any petrol pump, you will find that there is no greenery or no growth because of the leakage of the oil, petrol or other oils, oily substances, etc. in the nearby area. So can we have the recovery of that diversity which is present there? So this is one aspect which we used to study, that how to recover that one, that diversity, where the hydrocarbon degrading organisms, if we have the bioaugmentation with hydrocarbon degrading organisms, maybe eventually we can recover the diversity which is useful for the plant development, etc. So that was some project which earlier we had, but practically it was not checked. Now the last aspect, that how many minimum genes are required which are essential for the life. So minimum number is 206. So 206 genes are required, and uh, these are, and 382 genes are protein coding genes, may be essential in organism. But initially, these are the main metabolism aspects. We can see the DNA metabolism, 16 genes, RNA metabolism, 106, protein processing, etc., etc. So if can we have the synthetic organism? So that is one thing. And this person, Craig Venter, has developed one new organism, which is the synthetic one. He has transferred the genes from one organism to the another organism, and he has removed all the genes from that new organism, that is M. mycoides, and he has transferred the mm, uh, genes from the organism into this new organism, and found that this cell also grows well and with the different properties or different physiological traits. Of course, this creation has cost around $40 million to cause the one cell. But if it works, then it will be a good system in agriculture, where we can have the tailor-made microbial diversity for increased crop yield. And when and how, I don't know, but this is the concept which now people are trying to think, that can we have the tailor-made diversity for the forest development, for the development of the different plantation, and also in agriculture. Of course, the negative points we are not going to discuss here, but this is the future of the microbiology eventually. With this, I would like to thank Ajit and for giving me this opportunity. Dr. Santosh Tupe is my partner, and Dr. Vandana Gurmade, she is from Agriculture Institute. She is my student, but at present we are also collaborating on different aspects. And uh, so these three people have uh, given me a lot of support. Now, here one, just additional thing, that during COVID, we could not do much in our company. So we could publish 4 books on progress in mycology, and advancing frontiers in mycology and mycotechnology. And our recent book is on fungal biopolymers and biocomposite. All are Springer Nature book. Thank you so much for your patient listening. And if you have any questions, please let me know.
Yes. Thank you, sir. Uh, that was a wonderful, lot of interesting things, sir, that we learned. And before we start taking the questions, I just, you can all put your questions in the chat box, or if you want to ask them, you can open your microphone and ask directly to Deshpande sir. But before all of that, I think I just want to make sure that we thank all the people who made the talk today happen. And first of all, it's a heartfelt thanks to our learned speaker, Deshpande sir, for sharing his knowledge and passion with us today. Your insights have been truly inspiring. I think this has been a fitting talk in memory of Dr. V.D. Vartak sir, on his death anniversary. Vartak sir's legacy continues to inspire and guide us in our exploration as natural world as well as the sacred groves and their conservation. I must thank Ajit Vartak sir for initiating, coordinating, and driving things to make this talk happen. Also, thanks are due to Dr. Fatak as well as Fatak sir, who has been diligently and meticulously taking care of the technical side as well as the promotions and all parts of this session. Not to forget Shri Kaustub Mudgal, who made the attractive graphics that you all probably received along with the invitation for this talk. And I especially want to thank all those kind people who forwarded the information about this online talk so that it would reach a much wider world. We wouldn't have all the audience attendees without help of these people who have forwarded it. And finally, a big thanks to all each and every one of you who attended and joined us today. Your participation is what drives us to bring more such enlightening and insightful sessions to life, and can't wait to see you actually for our next session, which is going to happen very soon. What is it about? It's on 30th April, and the speaker is Shri Paresh Churi, and who will be speaking on botanical art. So that is going to be another interested one. So that information will be available through the WhatsApp message as well as through the website of at inher.in, that is inher.in, or Facebook page, as well as I mentioned, through the WhatsApp posts. So sir, I think we will open people if you have any questions. Feel free to open your mic and you can uh, ask directly to. There is one question that is in the chat box, sir, by Zabin Loni Moon, and she's asking: is there a particular microorganism pool that has been recorded as typical to the devrais of each region of India? Are these research opportunities available to students in conjunction with mentors per state?
Actually, this is very good question, and actually we wanted to start. Unfortunately, we could not do much in Maharashtra, but Meghalaya is one of the areas where you will find that the bioprospecting of Devrai microorganisms they are doing. And if anybody is interested, I mean, my email is there. They can write to me. I can send all the details about the bioprospecting of microorganisms from Devrai. So that is one aspect, and lot many things one can really do by using Devrai. I mean, just I gave the passing remark about Devrai, but certainly we can do that one.
Thank you, sir. I think, sir, there have been a lot of interesting concepts that you discussed in the talk, including right from cancer drugs to the diversity in what happens to the diversity between tropical forest and rubber plantations. Uh, so I think this whole field has a fantastic future. Which of these directions do you think holds most prospect in terms of changing human lives and our connection with our environment?
There are two different aspects. Is a changing human life is going for organic farming, because that is one thing which is according to me is important, and it can certainly give the eco-friendly technologies, which at present we are also doing that one, and because of that one all other problems will be solved. And so organic farming is one of the important thing, and that with using microorganisms we can go. And number of places we'll find that the wastewater treatment, etc. All these microorganisms are involved in that one, or the work which I have started with the gobar gas plant, etc. So these are the areas where microorganisms are playing role, and people are going. But unfortunately, that sort of awareness is not there. This is number one. And second thing is that I find that the researchers just publish paper and then just they forget. They are not, I mean, just to add, yesterday only I had written one article in Maharashtra Times about the startups. What are the issues and challenges in startups? So that is one thing where the researchers, though they are having very good publications, they are not thinking beyond that. If they think beyond that one, we can do really lot of wonders.
Right. You're absolutely right, sir. I think we right now we don't have that kind of ecosystem that we see existing in a lot of these Western
Hmm.
countries, where academia
Yes,
and the
yes.
businesses have good, what do you say, connections as well
Yes.
as good foundation. You know,
सर नमस्कार. मला एक प्रश्न आहे,
बोला
विचारू
मॅडम,
शकतो का?
बोला ना मॅडम.
सर आम्ही लोक गावामध्ये काम करतो आणि
हम्म.
म्हणजे मी स्वतः पण बॉटनिस्ट आहे, पण जे आज तुम्ही दाखवलं दॅट वॉज रिअली व्हेरी अमेझिंग मार्वलस. माझा एक प्रश्न आहे सर, की गावामध्ये आम्ही खूप वेळा लोकांना सांगतो की आपण जितकं पण फार्म वेस्ट आहे आणि काऊ डंक आहे त्याचं चांगलं मेन्युअर प्रिपेअर करू शकतो. काही
हो.
मायक्रोब्स आम्हाला ICAR नी दिले आहे, पण दे आर नॉट सफिशियंट. सर माझा असा प्रश्न आहे की आपण CN रेशो जर गाववाल्यांना सांगितलं तर ते त्यांना एवढं समजत नाही. एखादं कुठलं कॉम्प्लेक्स किंवा एखादं कंपोजिशन ऑफ मायक्रोब्स असं असेल ज्याच्याने की आपण CN रेशो तिथे डायरेक्ट मेंटेन करून चांगलं मेन्युअर बनवू शकतो, असं जर आपल्याला माहिती असेल तर प्लीज शेअर करा.
म्हणजे हा खूपच इलॅबोरेट हे आहे, परंतु असे मायक्रो ऑर्गॅनिझम जे आता तुम्ही म्हणताय की डिग्रेडेशनसाठी, थोडक्यात डिकॉम्पोजिशन ऑफ ऑर्गॅनिक वेस्ट,
या
ह्यासाठी जे काही मायक्रो ऑर्गॅनिझम लागतात त्याच्यामधलं मेन सगळ्यात आहे ट्रायकोडर्मा. ट्रायकोडर्मा जर तुम्ही वापरला तर सगळीकडे तुम्हाला ती वेस्ट ही चांगल्या पद्धतीने डिकॉम्पोज होऊन त्याचे सगळे न्यूट्रिएंट्स हे प्लांटला बेनिफिशियल असतात. त्यामुळे ट्रायकोडर्मा हा फंगस सगळ्यात महत्वाचा याच्यामध्ये. दुसरे ऑर्गॅनिझम्स आहेत बॅसिलस आणि सुडोमोनास, हे 2 ऑर्गॅनिझम. तसं म्हटलं तर 4 ऑर्गॅनिझम सगळ्यात महत्वाचे, ॲझोटोबॅक्टर, रायझोबियम, बॅसिलस, सुडोमोनास, हे 4 ऑर्गॅनिझम, आणि फंजायमध्ये ट्रायकोडर्मा हा फंगस. हे सगळे ऑर्गॅनिझम्स हे ह्या डिकॉम्पोजिशन मध्ये खूप उपयोगी पडतात. तर असं कन्सोर्शियम पण आपल्याला करता येतं आणि मिळतं. एक राहुरी कृषी विद्यापीठ आणि परभणी कृषी विद्यापीठामध्ये त्यांचे 8 ऑर्गॅनिझमचे कन्सोर्शिया आहेत अवेलेबल,
बर.
आणि
हा.
ते देतात रेग्युलरली.
ठीक आहे.
तर अशा प्रकारचा कन्सोर्शियम तुम्हाला रेग्युलरली मिळेल. आमच्या कंपनीकडनं पण तुम्हाला मिळू शकेल, पण आम्हाला आधी जर काही माहिती मिळाली आणि जर त्या संदर्भात काही जर तुम्हाला ऍडिशनल माहिती पाहिजे तर आम्ही त्याच्यामध्ये देऊ शकतो. परंतु राहुरी आणि परभणी विद्यापीठ हे दोघं कन्सोर्शिया बाजारामध्ये देतात, विकतात.
तसं सर आम्ही जे ICAR वाल्यांनी दिलं, डेव्हलप केलंय ते पण यूज करून पाहिलंय. तरी पण आमचे जे अगदी ट्रायबल बेल्ट मध्ये आम्ही कामं करतो तर तिथे ते खूप जास्त इफेक्टिव्ह मला असं दिसले नाही. म्हणजे तर का म्हणजे कारण ते काउडंग पण वेस्ट जातो, ते फार्म वेस्ट जे आहे ते पण वेस्ट असून
नाही,
राहिलंय.
काय सांगू का मी तुम्हाला, की सगळ्यात महत्वाचं म्हणजे तुमचा जो स्ट्रेन आहे, आपण त्याच्यावर आता काहीच बोललो नाही, पण
हा
स्ट्रेन
हा
हा
कुठला स्ट्रेन
चांगला
घ्यायचा.
पाहिजे.
हो
आता मी जे एक मेटारायझमचं एक उदाहरण दिलं मी, की कस्टर्ड ऍपल मधनं, आणि त्याच्या खाली एक मी टेबल दिलं होतं, एवढं मग सांगायला वेळ मिळाला नाही. माझ्याकडे स्वतःचे 40 मेटारायझम स्ट्रेन्स आहेत,
ओके.
वेगवेगळ्या, आणि ते वेगवेगळ्या होस्टवरनं आयसोलेट केलेले आहेत. म्हणजे याच्यावरनं हेलिकोवर्पा असेल, स्पोडोप्टेरा असेल, किंवा दुसऱ्या कुठल्याही पेस्टवरनं, याच्या व्हाईट ग्रबवरनं केलेले आहेत. तर ते असे जे आयसोलेट्स आहेत ते जास्त व्हिरुलंट असतात. आणि आता कस्टर्ड ॲपलचं आम्ही पाहिलं की कस्टर्ड ॲपलमध्ये इन्सेक्टिसाइडल ट्रेट्स असल्यामुळे, तसंच आम्ही कस्टर्ड ॲपल प्रमाणे नीम मधनं केलेलं आहे. नंतर दुसरं पपयामध्ये आहेत इन्सेक्टिसाइडल ट्रेट्स, आणि युकॅलिप्टसमध्ये, निलगिरीमध्ये पण आहेत. तर त्याच्यातही आम्ही आयसोलेट केले, आणि त्याच्यात पाहिलं तर त्यांच्यामध्ये इतके जास्त इन्सेक्टिसाइडल ट्रेट्स येतात. म्हणजे सांगायचा मुद्दा काय, की तुमचा स्ट्रेन हा चांगला इफेक्टिव्ह पाहिजे. नुसता ट्रायकोडर्मा घेतला म्हणून तो होत नाही. त्याच्यामध्ये सेल्युलेज ऍक्टिव्हिटी चांगली पाहिजे, हेमिसेल्युलेज ऍक्टिव्हिटी पाहिजे. लिग्निन डिग्रेडेशनसाठी तुम्हाला ट्रायकोडर्मा उपयोगी नाही, तुम्हाला स्पोरोट्रायकम पल्वेलेटम म्हणून ऑर्गॅनिझम आहे तो पाहिजे. आता ह्या ज्या गोष्टी आहेत, तसं न करता आपण एक ठोकताळे जे असतात ते देतात ऑर्गॅनिझम, आणि ते लॅबोरेटरी रिपीटेड सबकल्चरिंग मधनं त्याचं पोटेन्शियल गेलेलं असतं. कारण आपण लॅबोरेटरीमध्ये आर्टिफिशियल मीडियम वरती ते ठेवतो, की ज्याच्यामध्ये अतिशय रिच न्यूट्रिएंट्स असतात, त्यामुळे ऑर्गॅनिझम हॅपीली ग्रो होतो, आणि मग त्याची झगडण्याची प्रवृत्तीच निघून जाते
हम्म, हम्म.
आणि मग तो बाहेर चांगल्या पद्धतीने नेचरमध्ये वावरू शकत नाही. तर ते तसे स्ट्रेन्स, आता आम्ही स्ट्रेन जे मेंटेन करतो ते आम्ही इन्सेक्ट लार्वावरती मेंटेन करतो.
बरं.
म्हणजे त्याचं पोटेन्शियल जे असतं इन्सेक्ट पॅथोजेनेसिसचं, ते पोटेन्शियल मेंटेन राहतं. तर म्हणजे हा संपूर्ण वेगळा विषय आहे. मी त्याच्यावरती आम्ही पब्लिकेशन्सही आहेत त्याच्यात. परंतु तुम्हाला जर चांगले स्ट्रेन जर पाहिजे असतील तर तुम्ही ते आधी कन्फर्म करा की ते कमर्शियली प्रोड्यूस कसे केलेले आहेत, ते कुठल्या मीडियम मध्ये केलेत, काय केलेत, मग तुम्हाला चांगले मिळू शकते.
ठीक. खूप खूप थँक्यू सर. आणि मी आय रिअली रीच टू यू, कारण आम्ही 7-8 स्टेट मध्ये काम करतो ना, हा खरंच एक मोठं
मग तुम्ही
काम
आमच्याकडे जरूर या आणि आपण
हो.
त्याच्यावरती जास्त पद्धतीने बोलू. आणि आमच्याकडं नुसतं कंपनी म्हटल्यानंतर बाबा एक्स्ट्रॅक्शन ऑफ मनी असं नाहीये. आम्ही जास्तीत जास्त लोकांशी संवाद साधतो. आता मी ॲग्रोवन मध्ये पण लिहितो, तर संवाद साधला जातो शेतकऱ्यांशी आणि त्यांना जास्तीत जास्त मदत करणं हे जास्त महत्वाचं आहे.
थँक्यू व्हेरी मच सर. व्हेरी
ओके
इम्प्रेसिव्ह.
ओके, बाय.
थँक्यू व्हेरी मच.
थँक्यू.
सर, एक प्रश्न ॲक्च्युली चॅट बॉक्समध्ये होता, जबीनचाच परत होता, की शी इज इंटरेस्टेड टू शी वॉन्ट्स टू नो हाऊ डू वी शो इंटरेस्ट और कनेक्ट विथ पीपल हु आर वर्किंग अँड हेल्पिंग, हु वुड हेल्प हर विथ रिसर्च.
म्हणजे रिसर्च मध्ये म्हणजे त्यांना रिसर्च
या,
करायचा आहे.
हां, अँड शी वॉन्ट्स टू कनेक्ट.
हा, तर आता मला जर विचाराल तर सध्या आसाम ॲग्रिकल्चर युनिव्हर्सिटीमध्ये सगळ्यात जास्त चांगला रिसर्च चाललेला आहे ॲग्रिकल्चरमध्ये आणि मायक्रो ऑर्गॅनिझम्सचा. म्हणजे ते एक इन्स्टिट्यूट आहे. राहुरी अर्थातच आहे, परंतु राहुरी आणि परभणी या 2 ठिकाणी आपल्याला ॲडवाईस मिळू शकेल, नाही असं नाही. परंतु तामिळनाडू युनिव्हर्सिटी नंबर 1, आणि दुसरं IIHR म्हणजे ती याची बेंगलोरची इन्स्टिट्यूट आहे ती, आणि तिसरं आसाम ॲग्रिकल्चर युनिव्हर्सिटी, आणि ऑफकोर्स नंतर वरती दुसरी कुठली पंजाब युनिव्हर्सिटी. त्या अशा काही युनिव्हर्सिटीज आहेत की ज्या ठिकाणी तुम्हाला कनेक्शन चांगलं मिळू शकेल.
Right. Sir, there was this book which came out few years ago by author named Peter Wohlleben. I'm sure you probably heard of it. It's about trees and how trees were talking to each other through fungal network. And he mentioned one instance where, using that fungal network, they came to know about a tree which was in distress, which was cut few hundred years ago, and all the surrounding trees through that network continue to provide sufficient nutrition to keep that tree, although it didn't have any leaves or anything, but it kept it alive because they were part of this network, of this fungal network. So that was done in a temperate forest. And मला असं वाटतं की आय एम शुअर अवर फॉरेस्ट, ट्रॉपिकल सबट्रॉपिकल रिजन हॅव even more complex such networks, and which we probably lose out in the monoculture or those kind of plantations altogether, नाही का सर?
सर याच्याबद्दलचा आय मीन इम्पॉर्टंट सायन्स इज बायोमिमिक्री, and you will find that when the fungus grows, the mushrooms, they
Right.
always grow in a circle, and the fruiting bodies are in circle, but the mycelial networking is there, and that is one of the best examples to develop the drainage system in the colony. So biomimicry is one of the important aspect. And actually, I used to give a talk about how these organisms are important. And for example, याचं बुलेट ट्रेन, बुलेट ट्रेनचं जे तोंड आहे ते बीक याची किंगफिशरची बीक,
Kingfisher.
किंगफिशरची, आणि दुसरं त्याच्या वरचा जो हे आहे की ज्याच्यातनं हवा जाते ते दॅट इज मिमिक बाय फ्रॉम हे घुबड.
Right.
घुबडाच्या पंखाच्या याच्याप्रमाणे त्याच्यात जसे हे आहेत सेरेशन्स आहेत, त्या ज्याच्याप्रमाणे करून म्हणजे तेवढा त्या हवेचा रेजिस्टन्स कमी होतो, म्हणून बुलेट ट्रेन फास्ट जाते. तर हे जे आहे बायोमिमिक्री, ही सगळ्यात पण दुर्दैवाने आपल्याकडे काहीही डेव्हलप केलेलं नाही लोकांनी. अभ्यास इतके डायव्हर्सिटी आपल्याकडे आहे, पण अनफॉर्च्युनेट पार्ट इज दॅट वी आर नॉट युझिंग दॅट डायव्हर्सिटी फॉर द मॅनकाइंड. पण मशरूमचा आहे एक्झाम्पल, म्हणजे मशरूम कल्टिवेशन आणि नेटवर्किंग कसं होतं, आणि ऑल नेटवर्किंग इज विथ सेकंड मेसेंजर्स, दॅट इज द इंट्रासेल्युलर इफेक्टर्स, सायक्लिक AMP, सायक्लिक GMP हे 2 मॉलिक्युल्स. कॅल्शियम सिग्नलिंग मध्ये सगळ्यात कॅल्शियम इज इम्पॉर्टंट. सो ऑल द सिग्नलिंग इज डिपेंडंट ऑन दिस. आणि फॉर्च्युनेटली एक जस्ट ॲडिशनल थिंग्स होतो. आय वॉज वर्किंग ऑन द डायमॉर्फिझम. Rather, I still work on dimorphism. That is, a fungal organism changes its morphology as per the host environment. So, east to high fertility. So, that morphology is now connected to the cancer therapy. Morphological change कशामुळे होतो? So, what are the biochemical events? And that biochemical event is connected to the cancer therapy. And we have identified one enzyme, glutamate dehydrogenase. That enzyme is important in this morphological shift. So same enzyme can be used to develop the drugs. That means inhibitors of that enzyme can be used to develop the drugs, cancer drugs. म्हणजे इथपर्यंत त्याचा स्प्रेड आहे. परंतु अनफॉर्च्युनेटली आपण आता फोकस फक्त बघतो आणि आफ्टर पब्लिकेशन वी जस्ट फॉरगेट.
पण अमेझिंग आहे सर. आय थिंक देर आर लॉट ऑफ आन्सर्स. आपले health as well as environmental as issues
Yes.
we are facing, lot of these answers are in this research.
Yeah, and I want to find the answer for human health. Instead of mammalian cells, one should use yeast. म्हणजे तर वॅटसनचं हे म्हणणं होतं. वॅटसनना नोबेल प्राइज मिळालं होतं. त्यांचं
Watson and
असं
Crick.
म्हणणं, आणि त्यांना वॅटसनने असं म्हटलं होतं की बाबा, इफ यू वॉन्ट टू सॉल्व एनी प्रॉब्लेम अबाउट ह्युमन हेल्थ, इन्स्टेड ऑफ युझिंग मॅमॅलियन सेल्स यू शुड यूज ईस्ट. हे त्यांचं स्लोगन आहे. आता ईस्ट हा फंगस आहे. त्यामुळे सगळं मॉडेल सिस्टीम आणि 77% फिजिओलॉजिकल डिसऑर्डर्स आर स्टडीड युझिंग ईस्ट. मग खूप त्याच्यामध्ये स्कोप आहे.
Who would think, sir? I mean, it is as far away from humans, mammals, or vertebrates. एवढा लांबचं organism can
as a user model.
As a model is mind-boggling, sir. Sir, I would like to again heartfelt thanks for this wonderful lecture, sir, and giving us your time. Thank you so much, sir.
Okay, thank you so much, and I enjoyed that one.
Yes.
काय की जरा लाईक माइंडेड लोक असली समोर की जरा अजून उत्साह येतो.
नक्की सर, अगदी. आणि सर फार इंटरेस्टिंग विषय आहे.
आणि मला असं वाटतं की सरांची अजून ठेवायला हरकत नाही आपण फ्युचर मध्ये वेगळे विषय बायोमिमिक्री आणि सरांना सांगू असा अजून विषय घेऊन काय हरकत नाही. चांगला विषय.
एक पोस्टल स्टॅम्पचं मात्र करायचं आपल्याला. पोस्टल स्टॅम्प फंगल
ते करायचं. एक लेक्चर तुमचं आहे सांगितलंय, मायकोलॉजी ऑन स्टॅम्प आहे लेक्चर एक पुढे.
आणि बायोमिमिक्रीचं पण मी हे करीन. म्हणजे कशासाठी, नॉट फॉर काहीतरी दाखवण्यासाठी, पण लोकांना तो स्कोप अजून कळण्यासाठी की आपण काय काय करू शकतो. निवडुंगाचं झाड बघून दुबईला त्यांनी बिल्डिंग बांधलेली आहे. फक्त एरेशन कसं करत जातं आणि बाहेर गरम असताना आत एअर कंडिशनिंग कसं होतं. हे असे
तुमचा ईमेल आयडी बरेच मागतायत. ते ईमेल आयडी फक्त एकदा हे करा.
आहे ना याच्यावरती.
मध्ये ती स्लाइड किंवा परत दाखवलं किंवा
पहिली स्लाइडच आहे ना.
सांगितलं तरी हरकत नाही, सोपं आहे.
हो,
पहिली स्लाइड आहे. Mukund Deshpande 1952@gmail.com. आणि जरूर कॉन्टॅक्ट करा आणि आमची कंपनी बघायला या उरळी कांचनला.
सर ते फार आवडेल आम्हाला.
जरूर या. आणि आय विल बी हॅपी टू इंटरॅक्ट विथ अदर पीपल ऑल्सो.
चालतं नक्की सर. येस.
थँक्यू सो मच सर.
ओके, चालेल.
मी जस्ट ईमेल आयडी सरांचा टाकलेला आहे चॅट बॉक्समध्ये. सो यू कॅन जस्ट क्विकली कॉपी बिकॉज वन्स दिस सेशन इज एंडेड तुम्हाला त्या चॅट बॉक्सचं मिळणार नाही. इट इज अ व्हेरी स्ट्रेट फॉरवर्ड. सरांनी सांगितल्याप्रमाणे दोज वॉन्ट टू रीच आउट टू हिम, प्लीज नोट इट डाऊन.
आणि ईमेल वरती काही जरी असेल तरी मी तुम्हाला पब्लिकेशन्स म्हणजे जेवढं मी बोललो त्याचे सगळे पब्लिकेशन्स आर देअर विथ मी. सो पीपल कॅन सर्टनली सी दोज वन.
येस,
चालेल.
थँक्यू सो मच.
थँक्यू सो मच. ओके, थँक्यू.
थँक्यू. Thank you, sir.
Okay.
Just we will end it right here.
आणि
Thank you.
सर जाता एक कॉम्प्लिमेंट तुम्हाला द्यायची आहे. तुम्ही सुरुवात अतिशय सुंदर केली. म्हणजे मला असं वाटतं की ज्या टॉकची सुरुवात मी करायला पाहिजे ती तुम्ही ऑलरेडी केली होती.
Thank you, sir.
थँक्यू सो मच. आणि तुम्ही अतिशय आपली कधी गाठ पडली नाहीये अशी प्रत्यक्ष.
आणि आता गाठ घेऊ ना आम्ही तुमची.
आपण भेटूया आपण.
भेटू.
खरंच
चालेल.
छान वाटलं
Thank you,
मला.
sir.
म्हणजे
आवडेल.
ऑनेस्टली. हा जरूर. ओके.
Yes.
थँक्यू सो मच.
Thank you very much, sir.
Okay, thank you so much.
Yes, good to see.